Hello, I'm Marom Bikson. I'm excited to speak to you about how to optimize current delivery to the brain when doing transcranial electrical stimulation. These are my disclosures, and this is the link. You can scan it. That will bring you to a PDF of my slides if you wanted to consult them afterward. So my theme today is direction matters, and I think what I mean by that will become very clear as I develop this proposal. We have different forms of neuromodulation, going from technologies that require surgery for an implant, like deep brain stimulation, spinal cord stimulation. We have technologies like transcranial magnetic stimulation, or ECT, that are typically in-clinic treatments. And then we have technologies that we might call battery-powered or wearable, like transcranial direct current stimulation, that we imagine can go many places as well. And so we can think about which of these approaches is associated with the least cost, the least side effects, and the most deployability. And obviously, I think the TDCS and related technologies offer that advantage. However, when people think about targeting or where current flows to the brain, there's a perception that the direction goes in the opposite direction, where DBS and other technologies that are implanted are the most focal, whereas transcranial direct current stimulation and related technologies are the least focal. And I want to, through this talk, challenge that notion as well. So with conventional TDCS, we have to start thinking about initially pad-based TDCS. So this is TDCS using macroscopic electrodes. They're large, on the scale of a few centimeters by a few centimeters. And in 2009, we developed the first MRI-derived current flow models that predict how current travels through the brain during TDCS. And if you're not familiar with these models, you can look at that false color map on the side. And what you need to understand is that the red indicates regions of the brain that are receiving the most current, or we would say the most electric field. Green indicates regions of the brain that are receiving, let's say, a medium amount of current, and blue would be regions of the brain that are not receiving any current at all. They're not stimulated as well. So that's how you understand these pictures. And if you look at the prediction of what happens during TDCS, using a typical motor supraorbital montage, you can see that the current flow is very diffuse. It flows all the way from the one electrode, to the other, and along the way it passes through, so stimulates all brain regions along the way. And so you see that there's current not just under the electrodes, but actually really in between them as well. And this was a result of a computational model, but we and others went on to verify that those models were correct in this particular study. And again, I'll be putting all the citations below, and I'll just be discussing things now at a very high level. In this study by Huang et al., patients who were candidates for epilepsy surgery had electrodes implanted superficially along their cortex, and also depth electrodes, and transcranial electrical stimulation was applied, and the voltages that were generated inside their brain by the stimulation were recorded. The recorded voltages matched the model predicted voltages. And what that means at a very high level is it verified the models. It suggested that the prediction in the models that conventional TDCS produces very diffuse current flow were in fact correct. Now, in the same effort in 2009, we proposed something that went on to be called high-definition TDCS, which you can see, instead of using large electrodes, uses smaller electrodes and also can apply more than 2 electrodes on the head. Now, when we simulated the effect of doing this, we predicted that high-definition TDCS could be much more focal than conventional TDCS. You can see that the brain region now is sort of localized to under where the electrodes were. So why, what's the intuition on why that works? So let's first start with conventional TDCS. And large electrodes, there's a cartoon. We have 2 large electrodes on the head. One is called the anode and one is called the cathode. The anode is where current is pushed into the body, and the cathode is where current is collected out of the body. So we're pushing current in under the anode, and so it's flowing into the cortex. We are sucking it out at the cathode, and so it's flowing out on the cortex in that direction. You can see that the arrow direction has been flipped. And now we can maybe imagine that there is a neuron sitting under the anode, and there's a neuron sitting in the cortex under the cathode. And what happens to those neurons? Well, they polarize. Their membrane potential changes. Under the anode, if we imagine this sort of pyramidal, layer 5-6 pyramidal cortical neuron, what we know happens is that the soma depolarizes and the apical dendrite hyperpolarizes. So under the anode, we have soma depolarization, and this will sometimes be referred to as a nodal TDCS. On the opposite side, right, because the current is flowing in the opposite direction, we have a soma hyperpolarizing effect. The dendrite is depolarized, so soma hyperpolarized will be referred to as cathodal TDCS. And you have many cells, not just the ones, not just a few. And so we have many cells that are being either soma depolarized in red or soma hyperpolarized in blue. But unlike the way I've drawn the arrows here where they're just under the electrodes, the current has to go all the way from one electrode to the other. So it has to flow through all the intermediary brain regions and a bit wider as well. And so the actual pattern of current flow is much more like what is seen here. And what that means is we have a lot of cells that are being polarized all across the cortex, not just under the electrodes, but between the electrodes as well. And so that's giving us that very diffuse activation pattern with conventional TDCS. And again, this is just a direct consequence of how the current flows. What if we make the electrode smaller? Right? Will that solve the problem? And it will not. The reason is because the current still has to flow all the way from one electrode to the other electrode. So just making the electrode smaller doesn't solve the problem. We call these smaller electrodes high-definition electrodes, or HD electrodes. So that's where the term high-definition TDCS comes from. But what you're seeing here is just making the electrodes smaller, making them HD electrodes, does not guarantee focal current flow. It is not the size of the electrodes that matters the most, because current must still flow from one to the other. However, what we can do is move these electrodes closer together, and that does restrict the region of current flow, right? So now we can see that under the anode, current is flowing in, and it's quickly being sucked up by the cathode in the vicinity of it. And so making the electrode smaller and moving them closer together will result in a more focal neuromodulation effect. You'll notice, however, we still have a nodal, like excitatory effect, and a cathodal sort of inhibitory effect, right? So we still have this sort of push-pull going on even in this situation, right? And so when you have two electrodes, you'll always have to contend with the anodal and the cathodal region. So the next sort of trick that you can do is, instead of using one cathode electrode, imagine using two. And what that does is it takes that current that is being driven in under the anode and it splits it in two. And that means that under the anode, you have a stronger current flow than you do under the cathode. You've made the current under the cathode electrodes weaker. So we want to put a number on it. You can imagine if you're pushing 1 milliamp of current in, right, you are now splitting that and collecting negative 0.5 milliamp currents out on each one of the return electrodes. And so we are both more focal, but now also more unidirectional, because we only have the anodal effect now. And this can be extrapolated, if you look and think about the surface of the head, to having 4 return electrodes and 1 center one. And this is where we get the term 4x1 high-definition TDCS. 4 cathodes in this case on the outside, 1 anode on the inside. They're all small HD electrodes. And if we had, let's say, 1 milliamp coming in through the center one, we would be splitting the return current, in this case negative 0.25, across all the other ones. So 4x1 HD-tDCS, This is what it might look like on a head. This is a simulation of my head. You see the one anode in the middle, the 4 cathodes on the outside, but you have to realize that the cathodes are splitting the current, right? They're having less current in them than the middle electrodes, right? And we can use these simulations to predict how the current flows, and these are the results of those simulations. This is a little different though than what I showed you before. In this plot, if you look at the false color map, red means regions of a nodal where current is flowing in, excitation. Blue in this case means regions of cathodal where current is exiting. And green means there's no current flowing anywhere. And just what you can see by eye generally is you're seeing more red, right, than blue, and you're seeing that red is inside the area that's defined by the electrode. So not just focal current flow with 4x1 HD-tDCS, but also more unidirectional current flow, right? And that current flow region is defined by the ring. So the outer rings actually govern how much of the brain you will stimulate, right? It's not just under the middle electrode. It's not the size of the electrode again that matters. It is the circumference that is controlling how diffuse you would be. And so we can always assume that we are stimulating or passing current through the area of the brain that is under the ring. And we can actually make those electrodes come closer together, basically make the ring smaller. And by doing this, we can be more and more focal. So the tighter we make the ring, the more focal the brain stimulation is. There is, however, a cost where the tighter we make the ring, the more shunting there is across the scalp of current, and so the less intensity we have at the brain itself. And this can be shown, again can be illustrated with a computational model. In this case, you have 3 subjects. They're all getting 4x1 HD-tDCS. The only difference is, as we move across, is you can see that the size of the ring is changing from a 3 centimeter ring going bigger and bigger until a 7 centimeter ring. And you can see that in all cases, very robustly across all the subjects, right? As you make the ring bigger, right, you stimulate more and more region, and that the size of the region that you stimulate is the size of the ring. But what's also important is it's always reliably inside the ring, right? So whereas this is not necessarily the case with regular TDCS, with high-definition 4x1 TDCS, we can be very confident about where the current is going across subjects. It's inside the ring, and it's set by the size of the ring. And yes, as we make the ring smaller and smaller, there is a moderate reduction in resulting intensity, though of course you can always apply more current to compensate it. And so with 4x1, we can move it around, right? Not just over motor, we can put it over parietal regions, we can put it over frontal regions. And so the 4x1 HD-tDCS allows focal, cortical stimulation of any brain region just by positioning the ring over the targeted brain region. Usually we define that where we place the HD electrodes in terms of referencing EEG, 10-10 coordinates, because high definition electrodes are almost as small as EEG electrodes, and so we can describe where we're placing the HD electrodes based on the EEG system. So that's what it would look like on the head, where the light blue are the coordinates, and there are the electrodes of 4x1 placed over the front of the head. Now, here's a computer simulation showing that we can move the 4x1, right, from an anterior to a motor to a posterior position. And as we do that, the computer simulation predicts that the region of influence moves along with it. What we did in this study with Edwards et al. Is we didn't apply TDCS through these electrodes. All right, so here you have that anterior, right, the motor and the posterior situation, but we didn't apply regular TDCS. Instead, we used short 1,000 milliamp pulses. So TDCS is 1 milliamp or so. We're applying 1,000 milliamp pulses through these electrodes for a very short period of time. And when those electrodes were placed over the motor cortex, that large amount of current got into the motor cortex and it triggered an MEP, right? A motor evoked potential, a finger twitch essentially. At that same intensity, if we displace the ring, either anterior or posterior, and we zap again, we don't produce a motor twitch. So this was direct physiological proof that the 4x1 HD-tDCS, 4x1 montage in general, right, provides focal current flow. And there's much more nuance in this in the paper. One of the things we also did in this paper is we put TMS over the same region as the center electrode of the 4x1 montage. And as we displaced one, the electrical stimulation, we also displaced the magnetic stimulation. And at least at the resolution that we tested in this paper, we could not distinguish the focality between 4x1 electrical stimulation or transcranial magnetic stimulation, though which one is more focal ultimately will depend on the details of the magnetic stimulation and of the electrical stimulation. Now that was just acute effects. Michael Nietzsche and colleagues looked at the effects of 4x1 HD-tDCS on cortical excitability, and they used this using a standard measure where after the TDCS, transcranial magnetic stimulation is applied to produce a motor twitch. And the question is asked, did the TDCS make that twitch bigger, meaning an increase in excitability? Did it make that MEP smaller from baseline, meaning a decrease in excitability? And what they did in the study is they compared pad-based conventional TDCS, either with an anode over the motor cortex or a cathode, with 4x1 HD-tDCS, again, with either an anode or a cathode over the motor cortex. In both cases, the current was matched, and also the duration was matched, and it was only 10 minutes of stimulation. And what they found was that 4x1 HD-tDCS produced a change in excitability, as did conventional TDCS, but the magnitude and the time course of that change was very different. Whereas, for example, with conventional TDCS, the effects were no longer significant after about an hour. With 4x1 HD-tDCS, 2 hours. 2 hours after a 10-minute stimulation, the excitability changes were still significant, which is why they extended the experiment out for 6 hours. So 2 milliamps, just 10 minutes of 4x1 HD-tDCS over the cortex can produce at least 2 hours of excitability changes that are even manifest in the TMS MEP. So this showed this sort of lasting level of plasticity. And so with 4x1 HD-tDCS, I think what I'm trying to present to you is we have a tool that keeps the benefits of TDCS. It's low intensity, it's non-invasive, right? It's focal, it's deployable and everything else, but it has additional benefits, which is that it's focal, perhaps with comparable focality as TMS, and it's unidirectional, meaning the effects can be attributed to the polarity of the center electrode. And at this point, 4x1 high-definition TDCS has been used in hundreds of trials, including trials that have been focusing on figuring out the causal role of a particular brain region in an outcome. So if you want to get that kind of causality, you need focality, but also in several clinical trials where I think there's been suggestions that in certain situations, being more focal can actually increase efficacy. And I'm not going to be reviewing those trials here, but I wanted to bring your attention to just 3 of them that came out recently. 2 of them were run in Brazil, looking at both ICU COVID emergencies and also long COVID. Another trial that was run at UCLA using a cathodal variation. So now the middle electrode is a cathode for inhibition, for acute stroke management. And I think refer you to these trials that their encouragement, I think, speaks for themselves. But the trial I showed you by Michael Nietzsche and others was not the first trial on HD-tDCS. The first trial was actually by Igas, who's running this conference as well. And I want you to show you those results. So this was when he was working at the National Institute of Health. And I remember coming there in person and meeting with him and running this first trial of the effects of high-definition TDCS 4x1 on cortical excitability, again measuring TMS MEPs. And I'm going to refer you to the original paper for complete details, but what we found was a little bit puzzling. We were seeing what we believed were significant changes in excitability when we stimulated, but the direction of change, whether it was an increase or a decrease, was not always consistent across subjects. And this really puzzled us for a while, until once again, we consulted the computational models. And so what I'm showing you here are 3 models of 4x1 HD-tDCS, in all cases over the motor cortex. But what has happened is we've shifted the montage back and forward a very small amount, I think about 1 centimeter. And we've looked at what's happening at the motor cortex, specifically when it comes to directionality. And we found something very interesting about how small changes can lead to big changes in directionality that would explain this change in variability. So I want to explain that in some detail because I think it's very important. So here we have the 4x1 montage again with a deep, sort of intense and nodal stimulation and a reduced, and let's say this is the motor cortex, the hand knob. So we have a nodal stimulation of the hand knob, we have a surrounding inhibition region, and there is some inhibition there, right? It's not zero, right? It still has to come back. So there is an attenuated cathodal inhibition, but it's not zero at all. And so now I want you to imagine that instead of sort of a smooth cortex, right, which is not realistic in humans, we have this sort of highly folded, gyrated cortex. So now again, we have the hand area, and there's the anode sort of right over it, producing a nodal excitatory TDCS, but a periphery of cathodal inhibitory TDCS. What if we position the electrode a bit off? What if we slightly displace that ring? Not by much, right, but by half a gyri, right? You can see from this animation that if we're unlucky, a small displacement in position can lead to a categorical change in directionality. And we believe this was exactly what was happening in the original trial, just as a function of how we were positioning the electrodes. And so the models that you're seeing here were actually predicting directionality and showing us already that this could be a source of variability. Particularly, less so if you're looking at a target that's in the crown, but especially if you're looking at a target that might be on the gyri wall. So this is not a universal issue, but it's something to keep in mind with 4x1 HD-tDCS, that when directionality is critical and the target is in the wall, there's a possibility for variability. Now, one way to address this is to go back to a two-electrode system. So we're going to keep it as two electrodes, two HD electrodes. We're not going to move them that far apart. Specifically, we're going to position them across the target, right? So in this case, you can see the target remains the hand area of the motor cortex. We have two electrodes, and they're positioned across the target. And what you can see is that now the current is sweeping into a nodal, into the hand area, and exiting on the other side of the gyri. So we're placing it across the target. And in this situation, you can see that a potentially small displacement in the position of the electrodes, right, will not qualitatively change the direction of current flow, which gives us an extra level of robustness to the extent that we need it. So this is a very good situation which you really want consistent polarity across one gyri. That I want to mention, you now have to contend with the fact that you have the two polarities to work with. And if we think of the individuals with different shapes, different folds, right, in their head, right? So this is one individual, this is another individual. You can see that again, going across it, once again, gives us very reliable gyri wall polarization across different cortical folding, different sort of cortical morphologies. So again, using two electrodes gives us this wall level targeting. And so I'm calling this sub-gyri level targeting. It's not that the current is restricted to one gyri. It's that at one wall, one side of one gyri, we have precise control over how we are influencing that particular brain region. So this is this sort of, you would call it a 1x1, 1-to-1, one anode, one cathode, high-definition TDCS montage, right? Producing a consistent directionality of current across a wall. Now, these 1x1, two HD electrode montages have been tested for different reasons. This is a study by Rothwell and others, and I don't have time to fully explain it, but I'll refer you to the reference. But this study was very elegant, and they were looking at, again, 1x1 HD-tDCS. They were reversing the polarity, but also looking at direction, right? They were changing the flow to be not into the wall, but along the wall. A variety of magnetic stimulation was tested as well as sort of a probe. And the results were consistent with the theory that said that when you use a 1x1 montage like this, across the gyri wall, you can reliably polarize that wall in a way that changes excitability. It's very direction specific, but of course there is always more nuance, and I would refer you to the paper to studying that more. It was a very elegant, actually it's a series of neurophysiological studies using 1x1 HD-tDCS. So we have 4x1 HD-tDCS, unidirectional, broadly useful in studies where you really want to unidirectionally influence a brain region to understand its role in cognition and behavior and disease. But we have something else, which is this 2-electrode 1x1 HD-tDCS. The electrodes are close together, and this has been used in cases where there was a lot of interest in being very precise in how one gyri wall was being polarized. It's also not being used in clinical trials, for example, by Alex De Silva and colleagues, which have found that this kind of 1x1 wash across the gyri montage is remarkably effective in things like treating chronic pain. So those studies are emerging as well. So there's always a hand-in-hand neurophysiology, modeling neurophysiology and clinical trials. Now you can go back and actually take these 2 electrodes and move them farther apart, which is something I talked about earlier. And again, why would you want to go back to that? It turns out that doing that is very good if you want to stimulate the brain intensely. Now, to describe that, I just want to make a general point that when you're doing high definition TDCS, you have a lot of flexibility, right? Every location here in red could be a stimulating electrode, and each one of those stimulating electrodes could have a different amount of current, right? You could have +1, -0.3, +2, -0.4. As long as the currents add up to 0, you can stimulate with a tremendous amount of flexibility, well beyond just 4x1 or 1x1. So it's really a multi-electrode, or we might call it an MXN, M anodes and cathodes kind of approach. Many degrees of freedom. Now with so many degrees of freedom, you may get confused, I get confused. Well then where should I put the electrodes, right? Should I put them here? Should I put them here? How much current should I put in there? Fortunately, software has been developed. This is an example of some open source approaches that we have shared, which allows you to pick a brain region, right? You start with a brain region, and then you pick constraints such as my stimulator only has 4 channels on it, or my stimulator allows me to do 32 channels, and I only want to do 4 milliamp total current, and so on. You put the constraints in, and then the software automatically, in a closed form way, prescribes the optimal montage, meaning where you should put the electrodes, right? Maybe it prescribes 4x1 or something like it. Maybe it prescribes a 1x1 montage. But if you tell the software that my goal is to stimulate a brain region with maximum intensity, but I don't care about focality, I don't care what else I stimulate, I just want to hit that one brain region with maximum intensity, it will predict something like a 1 anode, 1 cathode at a far distance approach. And so what this paper teaches is a general idea that if you want to optimize for focality, you're going to have to compromise a little bit on intensity. And if you really want to maximize intensity, you're going to have to give up on focality. And so what a best montage is actually depends on what your goals are, which is why, again, a 2-electrode 1x1 HD-tDCS montage with distant electrodes, right, can make sense in certain situations where you want a lot of intensity, you still want your directional control, which can be down at the level of the gyri, right? And you don't necessarily care about being focal, meaning only stimulating one brain region. And again, you really have multiple options of what you can do with HD-tDCS. And I would emphasize, and including multi-target, you can go deep target, you can increase your intensity. So you can have, let's say, 3 anodal HD-tDCS electrodes, and you can put +2 milliamp on each one, so that's 6 milliamp. People like Benjamin Hamstead have been exploring higher intensities. A lot of potential flexibility with multi-electrode. In all cases, however, you can always be keeping in mind not just intensity across regions, but directionality as well, meaning direction matters. High definition, as I already mentioned, kind of looks like EEG, and in fact, it lends itself very well to be combined with EEG. So you have caps like this, which you show, in this case, the larger cups are the high definition electrodes, and the smaller points, the recording electrodes. It's very mechanically, functionally easy to combine HD-tDCS with things like EEG. And in fact, we and others have considered how you could use EEG to figure out where to stimulate. And so this will sometimes be referred to as reciprocity. And we've published a publication where, given a subject-specific anatomy, you can record the scalp voltages of a particular EEG response, and you can automatically, in a closed-form way, invert it using an algorithm in order to prescribe how you should be stimulating in order to stimulate the same brain regions that generate the EEG. And again, I will refer you to the paper for the full mathematical derivation of that. So you can see again and again how directions matter, right? Directions matter in the sense that how current flows through the brain, but also direction matters as in like how are we going to direct ourselves? What are the directions of how you're going to place electrodes, right? High definition TDCS, unidirectional, highly targeted across the cortex, maybe as targeted as TMS, maybe as targeted as other approaches. Proximal 1x1 for really gyri wall kind of effects, but more broadly, we have a lot of flexibility with high definition TDCS. And as we get to more and more electrodes, we really need software or EEG or other things to guide us, right? 4x1, 1x1, you can sort of use rules of thumb, not when you're talking about 32 electrodes. Now, I've talked about high definition TDCS, but I want to say that everything I say also applies for transcranial alternating current stimulation, transcranial random noise stimulation, and other things as well. But when you talk about TACS and TRNS, there's no sense of directionality in the sense that the directions, in any brain region, the directions are just kind of flipping back and forth all the time. But other than that, a lot of these rules do apply. And the final thing I want to go is I want to go back to conventional TDCS, because with conventional TDCS, there remains, first of all, a lot of promise as far as applications, obviously, but also still a lot of control and nuance in how the pads are placed and how they're optimized, including for gyri level directionality. So I have not talked about conventional pad TDCS, but I want to finish by saying a few words about it and how they can be optimized even at a gyri a specific level. So I want to go through an example where you want to target the left DLPFC. It's a common target, including in studies for depression. And let's say we want to excite it. So we're going to call this a nodal left DLPFC. And I want to describe an approach where you can use pads to optimize for a nodal TDCS. However, no subject-specific MRI, right? No neuronavigation. These are powerful but cumbersome tools to add. And moreover, I want to look at an optimization that allows you to place the electrodes very quickly and very simply without having to mess around with too much measurements. And so this approach, as often the case in my work, started with a computational model, but a constrained one. Again, we're going to be sticking to large pads, and we came up with this notion that you could have a headgear that holds the pads in place, right? So it automatically positions it. What can you control? You can control the angle of that bend, and you can control how far the electrodes are apart. So those are the two parameters we thought that you could adjust while still sticking with sort of like an automatic headgear kind of placement. I'll refer you to the paper. There are a lot of details here, but we're looking at conventional F3, F4, so placing the pads based on an F3, F4, other things as well, and something we call the OLE. And the OLE ended up being the superior approach for a nodal left DLPFC. Specifically, the OLE said that the angle should be 165 degrees, the distance, the inter-electrode distance should be 10 centimeters, and the model said that if you do it in this way, within these constraints, you optimize the amount of current that is arriving in the DLPFC, and also you optimize the amount of anodal or excitatory current that is arriving in the left DLPFC. That doesn't mean the current is stimulating only the left DLPFC. No, that wasn't the optimization problem. We didn't optimize for strict focality. We optimized for the excitation of the left DLPFC. And so this is this omnilateral electrode headgear. It's a montage, but the montage is really linked to a very simple to use automatic headgear. So that is the OLE montage. And it's been tried, including by Andre Brunoni and colleagues in their trial on TDCS for depression that was published now quite a few years ago. This trial reported that both antidepressant drug and TDCS were both superior, statistically superior to placebo, right, to the sham conditions as well. And they didn't just place the electrode sort of over the DLPFC. They didn't use the conventional F3 or 4. They did in fact use the OLE, right? So they used a optimized pad-based montage. And I only bring this up to emphasize that even with pads, there is nuance and it matters what you're doing. Even with pads and with high-definition TDCS, direction matters. And again, this is the link to my slides. And thank you for your attention.